everyone. Our project is Understanding Nanoparticle-cell Interactions to Improve Healthcare Diagnostics. I'm Megan Carey.
I'm Tanish Saikor-Vidamore.
I'm Sanyang Kim.
I'm Naranjan Shankar.
And I'm Bern Yi.
Okay, so first we're just going to give you a little outline. We're going to start off with some background on our topic, then move on to the procedure we used in our experiment and the results we drew. Then we'll give you our conclusions, future applications of what we learned, and then extensions on that.
Okay, so nanotechnology is the manipulation of matter on the nanometer scale. And just to give everyone an idea, a nanometer is 1/1000th of a meter. So nanotechnology has a lot of importance in the medical field. Current medical treatments such as chemotherapy and drug ingestion often deliver drugs indiscriminately. This is a problem because healthy cells as well as cancer cells can be attacked. However, using nanoparticles for medical procedures paves the way for non-invasive drug treatments, such as photothermal therapy and site-specific drug delivery. So in photothermal therapy, a nanoparticle in the bloodstream binds to a cancer cell and then penetrates the cell membrane. So once the nanoparticle is inside the cancer cell, a laser light of a certain wavelength is shined onto the nanoparticle. Then the nanoparticle converts this absorbed light into heat in a process called the photothermal effect. The cancer cell eventually dies due to the heat. In site-specific drug delivery, a nanoparticle carrying a drug will release that drug inside a cancer cell. So once the nanoparticle enters the cancer cell, certain chemical indicators inside the cell will bind to the surface of the nanoparticle, which then signals the nanoparticle to release the drug, which then destroys the cancer cell. So gold nanoparticles in particular are very suitable for biomedical purposes. They have optical and morphological properties that allow them to precisely monitor the chemical activities of the cells around them and also accurately target the appropriate cancer cell. Gold nanoparticles are also very good at converting absorbed light into heat, making them very good candidates for photothermal therapy. They're also generally non-cytotoxic, so they're less harmful to your body than nanoparticles of other metals. Finally, the gold surface of nanoparticles has a strong affinity for various biomolecules. So you could attach a variety of amino acids and proteins on the surface of the gold nanoparticle. This is important because you need specific surface ligands on the nanoparticle so that it doesn't get attacked by the immune system, so that it attaches to the correct cancer cell and penetrates the cell membrane, and also so that it releases the drug at the specific time and place. So this versatility of gold nanoparticles is very important for these non-invasive medical procedures.
So the self-assembly of these nanoparticles is a highly researched topic in the nanotechnology field. It has great promise because of the prospect of biomedical applications. Now when nanoparticles self-assemble, they usually self-assemble through mechanisms of intermolecular forces, such as London dispersion and dipole-dipole. However, when they agglomerate, they usually do so randomly, and these structures are not really functional. Therefore, we pursue the more complex directed self-assembly. And as the name suggests, directed self-assembly creates functional structures that could be used in the medical field. Now, in this specific project, what we did was we used computer simulations to simulate what we call nanoboxes. Nanoboxes are basically rectangular solids where each of the edges and all the faces are representative of nanoparticles. So the whole structure is composed of nanoparticles. These nanoboxes have extreme significance because they could be used for delivering nonpolar drugs to target the areas in the body, and also have been effectively shown to be perfect for photothermal therapy. And there's a couple case studies where this has actually fought breast cancer cells. So the goal of this project, the overarching goal, is basically to determine the characteristics that affect the self-assembly of nanoboxes and understand how this applies to the medical field. We did this in two parts. We did both an experimental part and a computational part. So now let's move on to the experimental part.
Okay, so to start off, we synthesized 12 nanometer gold nanospheres, and we did that by adding a gold salt stock solution into milky water, then heating it under mild stirring conditions. When it came to a boil, we added a sodium citrate solution, left it to boil for a little while, and then brought it back to room temperature and centrifuged the solution. We followed a very similar procedure to synthesize our 25 nanometer nanospheres. However, with this, we used a lower concentration of the citrate solution. So to synthesize our gold nanostars, we used a seed-mediated synthesis, meaning we took our nanospheres that we had previously synthesized and used them as a seed to create our nanostars using the reagent gold salt stock, silver nitrate, hydrochloric acid, and ascorbic acid. Okay, then to test for the size of our nanoparticles, we used a dynamic light scattering technique, which shines a monochromatic laser at the solution, then analyzes the light that scatters off the surface of the particles. Then we went on to dry a sample of it and test it under a transmission electron microscope to see the morphology of the particles. And each different type of nanoparticle has its own unique extinction spectrum. So to confirm the shapes of our nanoparticles, we centrifuged a sample of each solution and then tested it under a UV visible spectrophotometer. The extinction spectrum for the nanospheres showed peaks at 520 nanometers, and that confirmed that they were nanospheres. And the stars showed peaks at 520 nanometers and larger peaks at 800 nanometers, and that is characteristic of gold nanostars. And this, what you see here, is just a graph of the absorption spectrum of the 25 nanometer nanosphere. As you can see, there's a peak at the 520 nanometer wavelength, and that confirmed that we had created nanostars, or spheres, sorry.
Now here's a little bit of background before we move on to the computational aspect of our project. So anyone who's taken an introduction physics class or knows any basic physics principles knows that force equals mass times acceleration. Now for our computer simulations, this is very important because the force that actually drives these molecules to self-assemble is actually potential energy. So what Lennard-Jones potentials are is basically a representation of potential energy for non-bonding particles. So basically, here's a model of how Lennard-Jones potential works. As two particles are far away, they exhibit slight attractive force until they reach an equilibrium point. Now at this equilibrium point, the potential energy is the lowest that it will ever be, and the nanoparticles are very happy. They want to be there. But if they're pushed closer together, they'll exhibit a repulsive force on one another and then try to go back to equilibrium stage. Now, for the rest of the presentation, just keep in mind that negative potential energy denotes attractive force, while positive potential energy denotes repulsive force. And here's just a graph of the Lennard-Jones potential. On the x-axis is distance, and the y-axis is potential energy. Keeping in mind that negative potential energy is attractive, while positive is repulsive. As the distance comes closer together, there's more and more attractive force until it reaches an equilibrium point, and then there's repulsive force. And this is very important for the computer simulations that will be explained shortly.
So moving on to our computational procedure, first we created parameter-specific self-assemblies. And the specific parameters that we modified were volume fraction and nanobox dimensions. Now just to clarify, volume fraction means the total volume of all the nanoparticles within the simulation versus the total volume of the simulation size. And we use MATLAB in order to create these simulations. Now LAMS was the program that we used to run these simulations with these different parameters. Next, we used Visual Molecular Dynamics, VMD, in order to visualize the molecules that we had simulated. And as you can see on the right, there are cyan beads and pink beads. The cyan beads represent sticky nanoparticles, and the pink beads represent non-sticky nanoparticles. And these show how the nanoparticles are interacting with each other as they self-assemble. And next, we use this information in order to create graphs of the potential energy over time. So here we have our computational results, and we can mainly split these into two categories. One is the non-cube boxes, and second is the cube boxes. So the first important thing to note is the time at which a drop-off in potential energy first occurs. And this is because the sooner a drop-off in potential energy occurs, the faster the self-assembly rate is for said simulation. And as you can see here in these two graphs, none of the graphs of these non-cube boxes are overlapping. However, if we go on to the next slide, for the cube boxes, we do see overlaps between the volume fractions. As in the 4x4x4 particles, we cannot see the 0.005 or the 0.015. And this is because they have overlapped with another graph.
So let's talk about our conclusion. Our main question is, what are the effects of parameters on a rate of self-assembly? We use two variables, volume fractions and dimensional analysis. There's a positive correlation between volume fractions and the rate of self-assembly, and this is intuitive. The more particles you put in, the more often there'll be self-assembly. But what's really interesting is the overlaps, as Sandy mentioned. Only cubes had overlaps, and this means the data points are exactly the same for multiple graphs. Why is that? There's two ways of looking at it. You can say it's a limitation of the system. Our program is pretty basic and doesn't factor in all variables out there. Or we can also look at it this way. Nanocubes are optimal for self-assembly. Increase in volume fraction does not increase the rate of self-assembly because it is at its optimal state. On the dimensional side, we compared non-cube boxes to cube boxes, and cube boxes, again, are still the best at self-assembly. We could say definitely that cube boxes are what we should use for self-assembly. Finally, for optimal absorbance, as we mentioned before, 520 nanometers is the best optimal absorbance. I mentioned 520 nanometers again because of photothermal therapy. As we mentioned before, photothermal therapy is when gold nanoparticles enter cancerous cells and they're heated up to denature the proteins in the cancerous cells. Now we know the specific wavelength. We can shine a laser of 520 nanometers on these gold nanoparticles in these cells, and this causes minimal collateral damage to other cells. Optimal conditions for self-assembly were also found, and this is important not just for biomedical field, but also for other fields. Nanomaterials and nanoelectronics all need these kind of things, and our program is very malleable and can be functioned differently to solve these kind of problems. In the future, we want to use a more resource-intensive program. This kind of program could factor in a lot more variables, take a lot longer time, but also be a lot more detailed. We only use different parameters. We only use volume fraction and dimensional analysis, but we could also look at temperature. We also look at bead size, and these kind of things can give really interesting results and yield a lot of new information. And our final extension will be biological interactions. We are exploring the medical field. Cubes, nanostars, these kinds of things are brand new and haven't been studied much. If we add these things to cells, who knows what will happen? We could yield really interesting results and find a lot of new things. We will turn our theoretical knowledge into practical biomedical applications. We wouldn't have been able to do this project without the help of very special people. Dr. Laura Fabrice, Dr. Meenakshi Dutt, Rowan Conj, our wonderful RTA, Director Dr. Eileen Rosen, Associate Director Jean Patrick Antoine, and especially our sponsors, Rutgers City University of New Jersey, Rutgers School of Engineering, State of New Jersey, Civil Line Windows and Doors, Lockheed Martin, New Jersey Industries, Novo Nordisk Pharmaceuticals, Inc., and New Jersey Resources. Thank you for your time. We'll now open ourselves up to questions.
So these would be delivered, I don't know if you gave this info, intravenously into the body and then recognize only tumor cells?
So nanoboxes, if you're talking about nanoboxes themselves, they could be functionalized with ligands or something. And the method of inserting is really dependent on what you're thinking, but intravenous ingestion would be very possible.
Yes.
When you were doing your research, did you come across John Kanzi's research that he's using the gold particles with radio waves to kind of do something similar to what you're saying?
Sorry, what?
The John Kanzi's research, it's something to what you were saying. He used gold particles and radiation to do a similar thing where he treats cancer by inserting gold particles in, and then he uses radio waves to radiate the gold particles to kill the cancer cells.
That's interesting. I don't think we looked at that example specifically, but a lot of our literature research had to do with like the optical properties of gold nanoparticles. So I think that relates to what you were talking about with the radiation.
I did read a couple case studies where this has been effective in treating a good amount of patients. So yeah, there's literature out there.
When you did the experimental things, how do you know what you have at the end? Did you use a microscope? How do you know that you created the
That we
nanoparticles?
created the nanoparticles. Okay, so to test for that actual type of nanoparticle, the shape, we used what's called a UV visible spectrophotometer. And since each different one has its own unique extinction spectrum, it's going to have an optimal absorbance of light at a different wavelength. We tested which wavelength of light it absorbed, and this is a nanosphere, And since its optimal absorbance is at 520 nanometers, we were able to tell that that was a nanosphere.
And to add on to that, we also did dynamic light scattering and transmission electron microscopes. So that's like a multi-wave pronged approach to identifying our particles and making sure that we have this correctly sense them.
What were some of the greatest challenges you faced during the course of the project?
I think one of the main challenges that we faced was finding how to make the connection between experimental and computational. So that involved a lot of outside literature research. And then finally at the end, we were able to tie everything together and see how this applies to photothermal therapy and drug delivery.
What part of the spectrum is that wavelength? Like, is it visible? Is it infrared? What is it?
It's visible.
Yeah, visible light.
Visible light, yeah.
So how do you shine visible light into some cells inside of a body?
Yeah, actually, there's a whole field of optics of sending visible light into your body, especially like if you look at your finger, shine a laser through your finger, you can see through the other side. Once you get into deeper cells in this area, that's obviously one of the big challenges, but a more powerful laser could definitely reach the cells.
Thank you.
Yes.
Just putting what you've done in context, how much research is being done on nanoparticles and how much using gold? Was that unique to what you did, or is it just an extension of what's being done out there?
So gold nanoparticles have already been researched heavily, but the goal of our project was to find the optimal condition for self-assembly, because using gold three-dimensional structures that are made up of gold nanoparticles offers advantages that single gold nanoparticles don't offer.
Follow up on that, we ran simulation using nanoboxes, and nanoboxes could potentially be used to deliver non-polar drugs to targeted parts of the body, because they could essentially encapsulate a drug, and through ligands and antibodies and stuff like that, they can go to targeted areas and release the drug.
How do the particles differentiate between a cancerous cell and a normal cell?
So the gold nanoparticles are functionalized with certain surface ligands, so that way it does not bind to a healthy cell, because cancer cells will have certain surface markers that normal cells don't. So that's like an important aspect of site-specific drug delivery. And also the optical properties of gold nanoparticles allows them to monitor the chemical activities in the cells around them. So that's another way you can differentiate between a healthy cell and a cancerous cell.
How does the ligand bind to the surface of the nanoparticle? Is there a special end that has to be on the ligand, or is there a special way to get it to adhere?
And what's a ligand?
Yeah.
Okay, well, basically, how they bind is like basically just it has to have a certain structure, because if it doesn't have the appropriate structure, it won't be able to bind to the surface protein on the cancerous cell. So that's why like structure is very important for function and things like that. And in our project, like a ligand is basically just the surface material that will like bind to detect the cancer cell.
Any other questions?
Okay. If there aren't any more questions, thank you very much.